Hi, this is Eric Lyons, and this is a video tutorial on how to use CoGe's new system to load in your experimental SNP data. So what we're going to do is we're going to pull data from the 1001 Genomes Project, which is a catalog of Arabidopsis genetic variation. So you need to be logged into CoGe with a user account, and I'm currently logged in as the generic CoGe account, which we use for testing the system. And what you'll do is you'll go to My Profile page, which gives you a list of all your stuff in the system, as well as the ability to add new stuff. So what we're going to do is we're going to load an experiment. And so the Load Experiment page has a place for the name of the experiment, description, version, source, where these data came from, the genome to which they're associated, whether or not the data is restricted. What this means is it's only accessible to your user account until you share it with other users or make it fully public, and then the ability to actually load in the data. So these data are going to come from the 1001 project. What we're going to do is we're going to go to their data center, and we're going to pull data from this particular experiment, the MPIC Wang 2013. So I'm going to go into that data center, take a look at the releases, go to their current data, and strains underscore info. This is a metadata file, and what this is basically description about each one of the experiments. So for example, experiment 108, the name of the ecotype is LDV-18. It's from France. Here's the lat long positions, and Valerie Lecour collected it, as well as some additional information. So I'm going to go ahead and get that particular experiment loaded in. So I'm going to first copy the name of this particular ecotype, and I'm going to copy it in here for the name, and I'll say SNP data. For the description, version will be 1. Now these were done by the 1001 project, so I'm going to go ahead and select that. If that hasn't already been entered, you can go ahead and select a new one. The genome for this is going to be from Arabidopsis columbia 0, so I'm going to type in col0 and select one of these Arabidopsis genomes. This is the latest version. And now for the data files, I can upload it from my desktop, retrieve it from FTP site, or pull it in from the iPlant data store. We're going to get it directly from their website. So I'm going to leave this box open, come back over here and go to their strains, select strain 108, go to tear. Here is their VCF file for SNP data. I'm going to copy that link address, come back over here, paste it into the URL, and then say get. So now what's happening is these data are automatically being downloaded from the 1001 Genomes project page. It takes a little while for it to come in depending on the connection speed. 8 megs, not too bad. And when I'm done, click Load Experiment. Now what will happen behind the scenes is CoGe will decompress this file. It's been compressed as a GZ. It will try to figure out what kind of file it is. It's determined it's a VCF file. So now what it's going to go through is a process of validating that data file, making sure that it has the right number of columns and that every chromosome to which there's associated data, those chromosomes actually exist within the genome. Getting chromosome names correct can be sometimes a little difficult. Once that is done, it will index it. This is using FastBit for our data storage engine behind the scenes. And now it says it's all done. So now I can go to Experiment View. And this is CoGe's page for just taking a look at this specific experiment. So this is where I can make it public. I can launch the genome viewer, which I'll go ahead and launch and let that load in the background for a moment. But I also have the opportunity now to add annotations about this particular genome. So I can come back to the metadata file, and one of the things I can say for my annotation is that this is from the 1001 Arabidopsis Genomes Project, and specifically from the MPIC Wang 2013 data collection. I can come back over here. I'm going to just go back to the data center and go to this particular project page. I can copy the link to this, copy that in, and say for this type, this will be, data source. And I can go ahead and add that. And then this link will take me back over there. And I can do this for a bunch of the metadata. So the collector. That collector was Valerie. I can copy their name in, put it in. Oops, sorry. What I want to do is have that as the annotation, and the type is going to be collector. Go ahead and add that into the system, and I can keep doing that. as well as add images and so on and so forth. And I can change those, I can delete them, all those kind of things. Now I'm just going to look at the genome browser. So here's our track that we've just loaded in. These are SNP data. So the histogram is just showing you the density of SNPs if we're zoomed out too far. As we zoom in, we'll start to see individual SNPs. So each one of these colors represents a different kind of a SNP in terms of, is it an A to T, is it a C to G, is it a deletion? And as we continue to zoom in, we'll start to actually see the individual labels for what these SNPs are. Since this is integrated into the EPIC CoGe system, you have the opportunity, of course, to overlay additional kinds of information. So here's gene models. There's a bunch of experiments that we have in terms of methylation data, RNA-seq data, again, these polymorphism data. So you can go ahead and turn on these individual tracks and bring in some additional information. I'm going to zoom out a little bit so that hopefully we can see something in terms of some of these methylation marks around there. So here's our SNP trap, gene models, some methylation data. You can rearrange them and turn them off. So I hope that helps. If you do have any additional questions, definitely look at the page docs, our tutorials. Feel free to contact us or post questions to the CoGe forums. Thanks a bunch.